All right, so before we start today, there is a panel on belonging in STEM that is tomorrow evening in the Varsity Theater. So if you're interested in that, then please put that in your calendars. And then a reminder that the take-home quiz is due Friday in class, so make sure you bring that to class with you on Friday. And then our review sessions for this exam will be on Friday and Saturday. So today we're going to finish chapter 27. We're going to talk about three metabolic cycles, beta oxidation, glycolysis, and the citric acid cycle. And as I mentioned previously, we're not going to memorize every single step of the cycle. We're going to focus on the reactions that are occurring in each step and how they compare to reactions that we have learned previously. And in some cases, we'll learn mechanisms of these steps, but we're going to focus primarily on the chemistry as well as the purpose of each of these cycles and not necessarily memorizing every detail. So we'll obtain hopefully a working knowledge of these three cycles by the end of class today. So on Monday, we talked about how fatty acids or how triacylglycerols get converted into fatty acyl coenzyme A species. And then once that thioester is formed, then it gets chopped up 2 carbons at a time by the beta oxidation pathway, generating acetyl coenzyme A. Each cycle of the beta oxidation pathway will yield 1 molecule of acetyl coenzyme A and a fatty acyl coenzyme A derivative, another thioester that will have 2 fewer carbons than the original one. So the long chain fatty acids get degraded 2 carbons at a time, and those 2 carbons become acetyl coenzyme A derivatives. And so this is the pathway here on the screen. And the first step is an oxidation process. It's an oxidation because we're removing hydrogens. Anytime you reduce the number of carbon-hydrogen bonds, that will be classified as an oxidation. As we learned on Monday, oxidations of alcohols use NAD+. Oxidations of other functional groups are going to use FAD. And we went over that mechanism on Monday. It is a deprotonation to make an enolate, followed by an E1CB type of elimination, where this hydride, instead of being free and floating around in the active site of the enzyme, is just directly transferred to the nitrogen of the FAD due to the close proximity of the substrate and the coenzyme in the active site. Okay, so this is the mechanism. This is in fact that exact oxidation from the beta oxidation pathway, the formation of this double bond from the fatty acyl coenzyme A thioester derivative. So once that oxidation has been performed, this is an alpha beta unsaturated carbonyl compound. And we learned earlier on this semester that alpha beta unsaturated carbonyls are electrophilic both at the carbonyl carbon and at the beta carbon, so they can undergo a 1,2 addition or a 1,4 addition, also known as a conjugate addition. And in this particular case, a conjugate addition of water occurs, giving a beta hydroxy thioester derivative. And I've drawn the mechanism of that conjugate addition on the board here. So water is a weak nucleophile, which means we would need to deprotonate it before it can attack the beta carbon of our alpha beta unsaturated thioester. But because the active site of the enzyme has all of our reagents in very, very close proximity to each other, that deprotonation occurs simultaneous with the attack of the water on that beta carbon. And when we have nucleophiles performing conjugate additions, as we learned earlier on in the semester. That gives us an enolate intermediate, and that enolate intermediate simply gets protonated by an acid in the active site of the enzyme, giving the beta hydroxy thioester. Our enzymes, of course, are chiral. They're proteins. They're made up of L amino acids, and we're producing a stereocenter in this reaction. And that stereocenter is only produced as the S enantiomer. This is a stereoselective process in which only one enantiomer is formed, and that is because the active site of the enzyme is chiral. Okay. Any questions on how the conjugate addition step of our beta oxidation process proceeds? Okay, so once the conjugate addition has occurred, our next step is an oxidation of that alcohol to a ketone. And that process, as we learned on Monday, is going to be mediated by NAD+. Okay. And just as a reminder, we'll go back to the slide showing the NAD+ oxidation mechanism, where a base in the active site of the enzyme will deprotonate the OH, and then a hydride is transferred to the NAD+ molecule, to C4 of this NAD+ molecule, giving us this example. It's an aldehyde, and the example from the beta oxidation pathway, it's going to be a ketone. And then NADH, our reduced coenzyme, is produced in this process as well. Questions? Okay, so we'll go back to the cycle, the figure of the cycle. And so now that we have a beta keto thioester, a beta keto thioester is going to behave similarly to a beta keto ester. We learned back in I think it was chapter 18, where we learned about the Claisen reaction. We learned that beta keto esters are synthesized by Claisen reactions. And the reverse of the Claisen can also occur. And so a beta keto ester or a beta keto thioester can be broken up into two pieces via a retro-Claisen or a reverse Claisen reaction. And that's what happens here with coenzyme A. And we'll show you the mechanism of that fourth step in the beta oxidation process shown here. Again, hopefully this is becoming familiar to you now in the active sites of enzymes. We can deprotonate our nucleophiles in the same step as those nucleophiles attack an electrophile. We are able in the active site of enzymes to have three molecules come together simultaneously in this fashion. That generates a tetrahedral intermediate. Tetrahedral intermediates tend to eliminate leaving groups. In this case, the enolate is the leaving group that is eliminated in this carbon-carbon cleavage step. And that enolate is going to be a fairly basic species, of course, but the active site of the enzyme is designed to stabilize that enolate. There are presumably non-covalent interactions that are going to stabilize that enolate and promote its departure from this tetrahedral intermediate. And then, of course, there's going to be an acid in the active site of that enzyme that can protonate this enolate. And that gives the acetyl coenzyme A, and it gives our thioester where two carbons have been removed. Okay, so the purpose of this beta oxidation pathway is to turn our long chain fatty acid coenzyme A into acetyl coenzyme A, which can be dealt with in the citric acid cycle, as we'll learn later on this morning. And then also each cycle of the beta oxidation pathway is producing two reduced coenzymes. There are two oxidation steps in the pathway. There's the oxidation to form the double bond, and then there's the oxidation to convert the alcohol to a ketone. And in each of those oxidation steps, one of our reduced coenzymes is produced. And as we mentioned earlier, those reduced coenzymes are also energetic molecules through the electron transport pathway, they can release energy. Okay. So as I mentioned, each cycle is going to produce one acetyl coenzyme A and one of each of these reduced cofactors. So we can look here. Myristic acid is a C14 carboxylic acid. And so it is going to undergo a total of 6 beta oxidation cycles. The bonds in red are the ones that are going to be cleaved by those beta oxidations. And in all but the final one acetyl coenzyme A and one each of those coenzymes will be produced. And then in the final one, our C4 acyl coenzyme A, of course, will get cut into 2 acetyl coenzyme A's. So 6 cycles will produce 7 acetyl coenzyme A's, 6 FADHs, FADH2s, and then 6 NADHs. Okay. Any questions on the overall process of the beta oxidation pathway? Okay. If there are no questions, then I have a question for you. So let's get out your iClickers and go ahead and talk to your neighbors and place the steps of beta oxidation in the correct order. Okay. All right, let's try to get those final answers in. Okay, any more answers? All right, that looks like all for today. Okay, what happens first in beta oxidation? Oxidation, okay? What does that oxidation set us up for next? The conjugate addition. We form that double bond, it undergoes conjugate addition. Then what happens to the alcohol? Oxidation. And then our final step is our reverse Claisen reaction, breaking off our acetyl coenzyme A. Okay, so those are the steps of beta oxidation. The purpose of that is to generate those acetyl coenzyme A's, and then because there are 2 oxidation steps, we are also producing those reduced coenzymes. So now we're going to talk about glycolysis. So whereas the beta oxidation pathway is the catabolism of lipids, of triacylglycerols, glycolysis is the catabolism of carbohydrates, specifically glucose. So glycolysis is a 10-step pathway in which one glucose molecule is going to be converted into 2 pyruvate molecules. And because 10 steps is kind of lengthy, glycolysis is usually divided into 2 halves. And in the first half of glycolysis, we're going to take glucose and we're going to convert it into glyceraldehyde 3-phosphate. Okay, so we're going to essentially chop glucose in half. Glucose has 6 carbons. Glyceraldehyde 3-phosphate has 3 carbons. And so there's going to be a carbon-carbon bond cleavage. All 3 of the pathways of catabolism we're discussing today will have carbon-carbon bond cleavages. In the beta oxidation, it was the reverse Claisen. Here we will see it's going to be a retroaldol reaction. So Let's look at the steps in the glycolysis pathway. So the first step is a phosphorylation, where the primary alcohol of glucose is going to react with ATP, taking a phosphate. That's just going to be an SN2 type of a process, the alcohol attacking the phosphate on the end of the chain, giving us glucose 6-phosphate and ADP. Okay. And then we are going to, in our second step, isomerize glucose 6-phosphate into fructose 6-phosphate. Have we seen the isomerization of glucose to fructose before? We did. So what reagent did we use to isomerize glucose to fructose? We used base. We showed you that in the presence of a strong base, glucose can be isomerized to give fructose. It can be epimerized to give mannose. So this isomerization is similar but not identical to the one that I showed you previously with the strong base. This isomerization has fewer steps because, as we've learned by now, in the active site of an enzyme, it's possible to have 3 molecules reacting simultaneously. So this mechanism is easiest to see with the open chain isomers of these carbohydrates, right? They are in equilibrium with the open chain isomers. And so when you have the acyclic form of glucose 6-phosphate, it can undergo ketoenol tautomerization. In a solution, this tautomerization would require 2 steps, a deprotonation to form an enolate, followed by a protonation on the oxygen to form an enol. But in the active site of an enzyme, those 2 proton transfers can occur simultaneously, getting us directly to this enol. And the enol of a carbohydrate is a special type of enol. It is an enediol, having 2 OH groups on the alkene. So if we're going to tautomerize that enediol back to a carbonyl compound, we have 2 options because of the 2 OH groups. If we were to deprotonate here and protonate over here, we would just go back to glucose. But if we deprotonate the C2OH and protonate at C1, we've now converted or tautomerized to give fructose. So when we do this in solution, it takes more steps because the proton transfers have to occur individually. But in the active site of the enzyme, it just takes 2 steps to get from glucose to fructose. Okay? And then, of course, that open chain ketone is going to be in equilibrium with the cyclic hemiacetal form. Any questions about the isomerization of glucose to fructose? Okay, so going back to our cycle, once we have glucose 6-phosphate, we've now revealed another primary alcohol, and that primary alcohol undergoes a second phosphorylation reaction, consuming a second molecule of ATP. So you'll notice that in this first part of glycolysis, we are consuming ATP. We've consumed 2 molecules of ATP to phosphorylate our carbohydrate to give us fructose 1,6-bisphosphate. And that mechanism will just be the same as step 1. And what that sets us up for is our key carbon-carbon bond cleavage reaction that allows us to break our 6-carbon carbohydrate into 2 3-carbon molecules. And that retroaldol mechanism is shown here. So, and it's best seen in the open chain form. We have this equilibrium here. And the acyclic form of fructose 1,6-bisphosphate, because it has a beta hydroxy ketone. Anytime you have a beta hydroxy carbonyl compound, then a retroaldol reaction can occur. And in this case, we deprotonate that beta OH group at the same time as we're going to protonate this carbonyl. We're cleaving the carbon-carbon bond between C3 and C4. They've helpfully highlighted that bond that's being cleaved in red. And that gives us 2 species, an aldehyde, glyceraldehyde 3-phosphate. And then it also gives us an enediol. And as we saw in the glucose to fructose isomerization, that enediol can undergo a tautomerization. In this case, it undergoes tautomerization to give a ketone, dihydroxyacetone phosphate. Okay, so if we look at the structures of our carbohydrates in this, actually let's look at the structure on this slide because we'll see the acyclic version. So we could have a retroaldol. This is a beta hydroxyaldehyde. We could have a retroaldol reaction occur with the acyclic form of glucose 6-phosphate. We could deprotonate this OH, and we could have the enediol leave here, right? But that would give us a 2-carbon piece and a 4-carbon piece. And as you'll see later on in the cycle, we need to have 2 3-carbon molecules in order to perform the second half of glycolysis. So that's why it is necessary to isomerize the carbonyl from C1 to C2. By moving the carbonyl over 1 carbon, now we're going to cleave the C3-C4 bond in the retroaldol instead of the C2-C3 bond. Okay, so that isomerization is essential so that the retroaldol gives us 2 3-carbon molecules instead of a 4-carbon molecule and a 2-carbon molecule. Okay, any questions? All right, so once we have, let's go, okay, back to our cycle here. Once we've performed the retroaldol and we have glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, the second half of the glycolysis pathway requires glyceraldehyde 3-phosphate. So we have to isomerize dihydroxyacetone phosphate into glyceraldehyde 3-phosphate. Okay? How do you think that isomerization occurs? Is it going to be similar to a step we've already seen? Which one? It's going to be very similar to step 2, the glucose to fructose isomerization. We're going to go through an enediol intermediate. So it's going to be 2 tautomerizations, a tautomerization to form an enediol, and then a tautomerization from that enediol to the aldehyde. And so here's a figure showing that process. To a ketoenol tautomerization, and then back to a ketone. So these steps are, of course, reversible. Which of these 2 compounds is going to be more stable, a ketone or an aldehyde? A ketone. So if we were just relying on thermodynamics, this pathway would be in the direction of the dihydroxyacetone phosphate. But we need it to be the aldehyde. And so there's probably two reasons why we're getting the aldehyde in this case. One would be the active site of the enzyme is lowering the activation energy for the desired pathway to get the aldehyde. The second one would be Le Chatelier's principle, because dihydroxyacetone is not consumed by the second part of the glycolysis pathway, whereas glyceraldehyde 3-phosphate is. So the glyceraldehyde 3-phosphate gets consumed, and then according to Le Chatelier's principle, more of it would form. Okay? Any questions about our first half of the glycolysis pathway? Yes? Yes. Yes. Yes, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate are both produced in that retroaldol, and then dihydroxyacetone phosphate can be converted into glyceraldehyde 3-phosphate by this isomerization. So if we look at the entire pathway here, you can see we've consumed 2 molecules of ATP. Okay, so we've gone uphill in terms of the energy storage capacity of the cell in this particular case. And so it's in the second half. The second half is often known as the payoff portion of the glycolysis pathway, where we're going to actually produce molecules that can store energy. So let's take a look at that second half of the pathway. So the first step is an interesting one. So we're taking our glyceraldehyde 3-phosphate and we're oxidizing the aldehyde to the carboxylic acid oxidation state. We're going to form an acyl phosphate. And that acyl phosphate, of course, is one oxidation state higher than the aldehyde. Because the aldehyde is an oxygen-based functional group, we're going to use NAD+ for this oxidation. And I'll show you an outline of the mechanism of this process. It's an interesting mechanism. The active site of the enzyme that promotes this reaction is going to have a cysteine present in it, okay? And so the cysteine is going to attack the carbonyl, okay? So the cysteine in the active site of the enzyme attacks this aldehyde carbonyl, and then we're going to protonate the oxygen, and that will give us a tetrahedral intermediate looking like this. Okay, so this tetrahedral intermediate forms after the first step, nucleophilic addition followed by protonation of the oxygen. And then this is where the NAD+ comes in. I'm not going to draw out the NAD+ structure, but you can refer to it on the slide. We're going to have a base in the active site of the enzyme that can deprotonate the OH. The oxygen, hydrogen, electrons come down to form our carbonyl. And then our hydride is transferred to the NAD+ by the mechanism we've shown you previously. And so that's going to give us an enzyme-bound thioester. Okay. We form an NADH in the process. And then that enzyme-bound thioester is simply going to react with inorganic phosphate. This is not a transfer from ATP. This is just inorganic phosphate in solution that's going to attack the carbonyl and go from the thioester to the acyl phosphate. Okay. So any questions on this oxidation and phosphorylation step? Our first step of the second half. Questions? Mm-hmm. Yeah, it's going to be nucleophilic acyl substitution with the inorganic phosphate attacking the carbonyl and then the thiol functioning as the leaving group. Yes?
I know that yesterday we talked about the fact that NAD+ mainly oxidizes alcohols.
Uh-huh.
So is this kind of another thing that it does? It's helping with this oxidation?
Yes and no. So it's oxidizing an aldehyde, but you can see the intermediate has an OH. So it's still oxidizing alcohols. And it's kind of like what we saw earlier in the class when we learned, actually it was last semester in chapter 11, when we learned about chromium oxidations. To oxidize an aldehyde, you first have to convert it to a hydrate where you have OH groups present. So this is kind of analogous to that hydrate intermediate that we would form in solution from an aldehyde. Okay. All right, so then we have another phosphorylation step. Okay, so we're consuming, we're transferring a phosphate in this step. The acyl phosphate is able to transfer the phosphate to ADP, and we'll show you the mechanism of that step here. It's basically an SN2 type of a step where the terminal phosphate of ADP is attacking the phosphorus, and the carboxylate is just the leaving group. So we've seen acyl phosphates behave in two different ways. We've seen acyl phosphates get converted to thioesters. That's what happens before the beta oxidation pathway, where we take a fatty acid, we convert that fatty acid to an acyl phosphate, and then it goes to a thioester. So nucleophiles can either attack the carbonyl of an acyl phosphate, or they can attack the phosphorus atom of an acyl phosphate. And which occurs will depend on the nature of the nucleophile. It will depend on the nature of the active site of the enzyme. In this particular case, we're using the acyl phosphate not as a carboxylic acid derivative for nucleophilic acyl substitution. We're using it as a way of transferring phosphate to ADP. Okay, so this transfer gives us a molecule of ATP. Okay, any questions? Okay, so let's go back to our cycle. now we have a different kind of isomerization occurring, which is transfer of a phosphate from C3 to C2. And that is not a one-step process. It's not an intramolecular process. It's actually an intermolecular process. You're going to have phosphate end up here, and then you're going to take that phosphate away. So we're not really going to go over the mechanism of how that happens. But that sets us up for step 9, our next to last step in the pathway, and that is a dehydration reaction. Okay, so this is a beta hydroxy carbonyl compound. Where have we seen dehydrations of beta hydroxy carbonyl compounds before? Aldol reaction, right? Aldol reaction forms beta hydroxy carbonyl compounds, and then under some conditions, they undergo dehydration to give alpha beta unsaturated carbonyl compounds. And what is the mechanism of that reaction? It is an E1CB mechanism that goes through an enolate intermediate. And that's what happens in this step is we have an E1CB. We're going to form an enolate. So let me make sure I get my stereochemistry right here. And my phosphate. Okay, so this is our intermediate, our 2-phosphoglycerate that we have here. We're going to have a base, deprotonate. We don't often make enolates of carboxylic acids because, or carboxylates, because those carboxylates are already negatively charged. But the active site of the enzyme is going to lower the activation energy for this pathway. And then once we have this intermediate enolate, then we're going to have elimination of the OH, but rather than form OH, when that comes off, it's just going to take the proton from the base, and that's going to give us our phosphoenol pyruvate and reform our base. Okay, so it's an E1CB mechanism via an enolate intermediate. And then we don't have free hydroxide floating around. It's just going to get protonated as it is lost from the molecule. Okay. Any questions about this dehydration mechanism that we see? Okay. And then that's going to set us up for the final step of the glycolysis pathway, and that is a second phosphate transfer from phosphoenol pyruvate to ADP. And that's going to proceed very similarly to the first phosphate transfer that I showed you, just an SN2 type of a process. In this case, instead of a carboxylate being the leaving group, it would be an enolate, but that enolate gets protonated as it leaves. So you displace the phosphate group from phosphoenol pyruvate, and that gives us pyruvate, our 3-carbon molecule that is the product of glycolysis, and it gives us ATP. Okay, any questions about that? So if we look at the cycle, or the second half of glycolysis, we need to remember that 2 molecules of glyceraldehyde 3-phosphate enter this for every 1 molecule of glucose that we started with. Okay, so how many molecules of ATP are produced from 1 molecule of glucose? We're going to produce 4. How many did we consume in the first half? We consumed 2, so the net result is we've produced 2 molecules of ATP. And then how many molecules of NADH do we produce for 1 molecule of glucose? We're going to produce 2. So the net result, if we want to draw the net equation for glycolysis, we're starting with glucose. And we are using 2 NAD pluses. We're going to convert that into 2 molecules of pyruvate. Pyruvate is our alpha keto carboxylate species. We've produced 2 molecules of NADH, 2 protons that are just transferred to various spaces. And then we're going to have 2 ADPs converted into 2 ATPs. So this is our net equation for the glycolysis pathway. Okay? Any questions about that? Yes?
Is this how most life produces ATP and NADH, or is this just how?
As far as I know, yes, this is a very common pathway in nature. I'm not sure exactly what organisms use it and what don't, but certainly we use it, and I think it's common amongst other animals as well as a way of metabolizing glucose to produce ATP and to produce NADH. So then we have to consider the fate of the pyruvate. What happens to the pyruvate? And that depends on the conditions. It also depends on the type of organism. So pyruvate can be converted into acetyl coenzyme A. This is going to happen under this happens under aerobic conditions. Okay. Or it can be converted into lactate. Lactate, the conjugate base of lactic acid, that happens under anaerobic conditions. Or in organisms such as yeast that perform fermentation, ethanol can be produced. Okay, so the fate of pyruvate depends on the type of organism as well as the conditions. So let's take a look. Pyruvate to acetyl coenzyme A. This is going to be an oxidation process. We're oxidizing the ketone carbonyl. We're decarboxylating. We're losing CO2 in this process. We're not going to go over the mechanism here, but it is an oxidation of a ketone, so it may be similar to that oxidation we showed you that we have now up on the top board here of the aldehyde, but we end up with the thioester. So this occurs as long as there is sufficient NAD plus, right? Even though NADH is the molecule that is used for energy storage, the reduced coenzyme, we have to have enough NAD plus in the cell to perform glycolysis. If you look at our net equation of glycolysis, it requires 2 molecules of NAD plus for every 1 molecule of glucose. So even though NADH is an energy storage molecule, we have to have NAD plus as well. And so the body has a pathway for oxygen turning NADH into NAD plus. So if there's sufficient oxygen present, you're going to have a high enough NAD plus concentration that you can turn pyruvate into acetyl coenzyme A. And then acetyl coenzyme A will get metabolized via the citric acid cycle, which we will see in a moment. Okay? Under anaerobic conditions, your pyruvate's going to get converted to lactate. And the entire purpose of this pathway is to generate more NAD+. So if you're exercising, if your muscles are using glycolysis to produce energy, and if you've depleted your NAD+, if you don't have much NAD+ in your muscles, then your ability to keep working out, keep playing your sport, keep doing whatever you're doing is going to diminish. And so your muscles are going to need that NAD+. This is a way for your muscles, if there's not enough oxygen, if you're not getting enough oxygen in to recycle that NADH to NAD+, your body can use this pathway. It can reduce pyruvate using NADH to produce lactate and NAD+. And now that NAD+ is available to continue breaking down glucose via the glycolysis pathway. Okay, question.
What happens to the lactate after it's produced?
So the lactate will build up in your muscles, and that's what happens when your muscles are sore. If you've ever heard people talk about getting lactic acid, having too much lactic acid, it's sore muscles from overuse, and then that that lactate eventually will be after exercise is over, then this process can be reversed. All right. So if you have increased oxygen, then you're going to have enough NAD plus to reverse this and go back to pyruvate. So it's just something that's temporarily performed in order to get NAD plus. Yes.
So why does having lactate in your body make you sore?
I don't know the answer to that. I'm not sure. But it's good that we have that, because then that's a sign that perhaps we should take a break and give our body a chance to produce more NAD plus, so that we can then undergo the regular glycolysis pathway. Okay. And then the final one. This is what happens in yeast. Pyruvate to ethanol. This is going to be a reduction. Okay. A reduction. So that's going to use NADH. And remember, back in 351, we talked about ethanol and how ethanol is produced by fermentation processes, the fermentation of various carbohydrate-containing materials, fermentation of barley, fermentation of grapes. So you take grapes or barley, you mix them with yeast, and you get ethanol by this process. So this is how alcoholic beverages are produced by yeast. Okay? So a lot of different things can happen with pyruvate. But what about the acetyl coenzyme A? So the acetyl coenzyme A that is produced then goes into the citric acid cycle. Okay, so here's our final cycle that we're going to study today. And this is a cycle that begins and ends with oxaloacetate. So this oxaloacetate molecule is going to be the starting point. It's going to react with acetyl coenzyme A, and then we're going to go through the whole cycle, and we're going to get back to oxaloacetate. So the purpose of this cycle is to metabolize or consume our acetyl coenzyme A. And in our two other cycles, our two other catabolism pathways, we've had carbon-carbon bond forming reactions, or carbon-carbon bond cleavage reactions, sorry. We've cleaved a carbon-carbon bond in beta oxidation by a reverse Claisen. We've cleaved a carbon-carbon bond in glycolysis by a retroaldol. Here we're going to cleave carbon-carbon bonds of the acetyl coenzyme A by decarboxylation. So decarboxylation is our third type of carbon-carbon bond cleavage that we will see in these pathways. And so the purpose here is to convert the carbons of acetyl coenzyme A into CO2. CO2 is very easily excreted. It just is exhaled in our breath. And then in that process, we're going to generate reduced coenzymes. So those are energy storage molecules. And we're going to produce GTP, which is a close relative of ATP. It just has guanosine instead of adenosine, just a different heterocyclic base. But we're going to produce a GTP in this pathway. Okay? So again, we're going to go through these steps similar to how we went through glycolysis and beta oxidation, focusing on the mechanisms of steps and comparing them to reactions that we have studied previously. So first step is going to be an aldol. We have to do a carbon-carbon bond forming reaction to attach our acetyl coenzyme A to the oxaloacetate molecule. And that's going to go first by formation of an enol. So we have simultaneous proton transfers to form an enol, and then that enol attacks the ketone, and then that gives us the aldol product. Here's the bond that was formed in red. And that thioester was used to make it easier. Well, we had the thioester because it was the product of the beta oxidation pathway, and we can get it from pyruvate, of course. But that thioester now is then going to be hydrolyzed. This is just going to be a nucleophilic acyl substitution with water being deprotonated as the nucleophile, giving us citrate, or the conjugate base of citric acid, the molecule that gives the cycle its name. Okay. Any questions about this aldol reaction? Okay, so then the next step in the process is going to be an isomerization where we're going to move the position of an OH group. So this is going to be different from our earlier isomerizations. We've seen moving the position of a carbonyl. We've seen moving a phosphate. This is a new one. We're going to be moving the position of an OH group. And this is pretty neat how this occurs. It's going to have two parts to it. We're going to dehydrate this OH. What mechanism do you think we're going to use to dehydrate? E1CB, exactly. This is beta to two different carbonyls, right? So it doesn't matter which one. We would dehydrate and we get the same intermediate. And once we've dehydrated, we can rehydrate. That's going to be the conjugate addition mechanism that is right here, where we're going to perform conjugate addition of OH here. And again, we're going to get a single isomer, single enantiomer of this product, okay? So dehydration followed by conjugate addition is how that occurs. So once we have the isocitrate molecule, that isocitrate molecule is going to undergo NAD plus oxidation. That's a step we've seen multiple times this morning. And that gives us a beta keto carboxylate. And we know that when we have 1,3-dicarbonyl compounds, those can undergo decarboxylations. We learned that back when we were studying the malonic ester synthesis, acetoacetic ester synthesis. And so here we have a biological version of that decarboxylation, slightly different mechanism, because we have a carboxylate instead of a carboxylic acid. So that's going to decarboxylate to give an enolate that then just gets protonated, okay? So that's how we lose one CO2 molecule, produce one CO2 molecule, and we've produced an NADH in the process because there was an oxidation that occurred, okay? Any questions? So once we have the alpha ketoglutarate, the next process involves decarboxylation and oxidation. We're not going to go over the mechanism of that process. It's going to be similar to what happens with pyruvate. If you look at the conversion of pyruvate to acetyl coenzyme A, we've taken this ketone and turned it into a thioester. And so that's very similar to what's happening here. We're taking this ketone and turning it into a thioester with loss of CO2 and production of NADH. So we're not going to go over the mechanism here, but we're just going to compare this to what happens in the conversion of pyruvate to acetyl coenzyme A. All right, so then once we have our thioester, that thioester can undergo nucleophilic acyl substitution. We've seen that before. The formation of an acyl phosphate. We saw that in the glycolysis pathway. And then we're going to transfer that phosphate to a GDP in this case instead of an ADP, but the purpose is the same. Same mechanism that we showed you in the glycolysis. So that's going to give GTP. That's going to give succinate. Okay. And then once we have succinate, we're in the home stretch of the cycle here. We're going to oxidize succinate by forming a double bond. Where have we seen this step before? Fatty acid beta oxidation. So it's the exact same mechanism. We're going to deprotonate to form an enolate, and then that enolate is going to eliminate a hydride, transfer a hydride to the FAD. So it's that same mechanism we showed you at the beginning of class today to get fumarate. And then, just like beta oxidation, we're going to have a conjugate addition of water to that double bond to give malate, and an enantioselective conjugate addition, followed by an oxidation, NAD plus mediated oxidation. So you'll see that the last three steps of the citric acid pathway are the same as the first three steps of beta oxidation. Okay, we're doing an oxidation, a conjugate addition, and another oxidation. Okay? So if we look at the net equation, you can see that for each acetyl coenzyme A that goes through the cycle, we get 2 molecules of CO2. We get 1 molecule of GTP, that's an energy source. And then we get 4 reduced coenzymes because of the 4 oxidation steps that we have in the pathway that can then transfer electrons via the electron transport chain. Okay? So we can calculate, we're not going to calculate this specifically, but we'll just show you the results of the calculations. We can calculate the total ATP yield from 1 molecule of glucose because we know how many ATP equivalents are produced by our reduced coenzymes. And we find that the total ATP yield, when we combine glycolysis with the citric acid cycle, that total ATP yield is 32 for 1 molecule of glucose. Okay? But, yeah, question.
How long are we gonna have to know both of these processes? Do we need to know names, reagents?
Working familiarity is what I would say. Understanding mechanisms of reactions we've learned, being able to predict products given one of those intermediates, but not reproducing the entire pathway. But understanding the purposes of these pathways also. Those are the things we're focusing on. So last thing for today is that even though it seems impressive, we're getting 32 molecules of ATP from 1 molecule of glucose, the ATP yield from beta oxidation combined with the citric acid cycle is even more impressive. So 1 molecule of lauric acid is going to give us 78 ATPs. Now there are more carbons in lauric acid than there are in glucose. So you can divide the ATP yield by the number of carbons in the molecule you start with, and you find that the beta oxidation pathway does come out ahead. You get about 6.5 ATPs per carbon, whereas in glucose, you're getting about 5.5 ATPs per carbon. So that's why lipids are indeed a slightly better source of energy than carbohydrates. All right, that's it for chapter 27. We'll start chapter 30 on Friday.